Imagine having the power to eliminate malaria, a disease that still kills over 600,000 people annually. Not with drugs or vaccines, but by simply changing the DNA of the mosquitoes that transmit it. Imagine pressing a genetic delete button on invasive rodents, devastating island ecosystems, or by permanently modifying disease-carrying ticks. This isn't science fiction, this is the promise of gene drives. But here's where it gets really wild. These genetic changes wouldn't just affect individual organisms, they would cascade through entire wild populations, potentially eliminating or fundamentally altering species that have existed for millions of years. It's like having an edit button for the planet's genetic code. The power is staggering, the potential benefits enormous, and the ethical questions, well, they're keeping bioethicists up at night. So what exactly is a gene drive? At its core, it's a genetic technology that dramatically increases the odds of a particular gene being passed to offspring, breaking the conventional 50-50 rule of inheritance that Mendel discovered in his pea plants. Instead of a coin flip determining whether a genetic trait gets passed down, gene drives effectively load the dice, ensuring nearly 100% inheritance rates. This creates a genetic wave that can sweep through an entire wild population in just a few generations. The concept of gene drives has actually been around for decades. Back in the 1960s, evolutionary biologists theorized about selfish genetic elements, bits of DNA that could bias their own inheritance. But these remained largely theoretical until a revolutionary technology called CRISPR-Cas9 came along around 2012. CRISPR gave scientists the precision tools needed to create gene drives in the lab, turning a biological curiosity into a powerful technology. The first proof of concept gene drives appeared in fruit flies and yeast around 2015, and since then, development has accelerated dramatically. We now have working gene drives in malaria mosquitoes, which could be our first real application in the wild. In 2023, researchers announced the first successful gene drive in Culex mosquitoes, vectors for West Nile virus and other deadly diseases, adding yet another species to the growing list of potential targets. But here's where the story gets complex and why we're dedicating this entire episode to the topic. When we talk about gene drives, we're not just discussing a neat laboratory technique, we're contemplating a technology that could permanently alter the biosphere in ways we might not fully understand. The promise is undeniable. Diseases like malaria, dengue, and Lyme could potentially be drastically reduced or even eliminated from the world. Invasive species that devastate ecosystems might be precisely controlled without toxic chemicals. Agricultural pests could be managed with fewer pesticides. But like Uncle Ben says, with great power comes great responsibility. Once released, gene drives are designed to spread. That's their purpose. They don't respect national boundaries, consent forms, or regulatory frameworks. They raise fundamental questions about who gets to decide which species get modified or potentially eliminated. How can we predict the ecological ripple effects and whether we can truly control such a potent technology once it's unleashed? Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're exploring gene drives, a technology that could literally rewrite the rules of evolution. Gene drives could be leveraged to finally eliminate some of the largest killers and transmitters of disease on the planet. But if they are not used properly, they could cause ecological catastrophes, unforeseen detrimental effects, or could be used to bring out the worst in humanity. We'll explore the science behind gene drives, their potential applications, the manufacturing challenges in scaling this technology, and the complex ethical considerations they raise. We'll examine the safeguards being developed and the governance frameworks being established to ensure responsible use. Because make no mistake, gene drives represent one of the most powerful technologies humans have ever developed, with the capacity to reshape our relationship with nature and disease. The question isn't just can we do this, but should we? And if so, how do we proceed wisely? Let's break down how gene drives actually work, because the science here is both fascinating and crucial to understanding their potential impact. In normal sexual reproduction, each parent contributes one set of genes to their offspring, giving each genetic trait a 50% chance of being passed down. It's like flipping a coin. Heads, you get your mom's variant of a gene, tails, you get your dad's. This 50-50 chance is why harmful genetic mutations typically don't spread through populations easily. Natural selection weeds them out over time. Gene drives fundamentally hack this system. Instead of accepting the coin flip, they actively convert the other parent's gene to match their own, effectively changing that 50% inheritance chance to nearly 100%. Imagine a coin that no matter how you flip it, almost always lands on heads. The most common method for creating gene drives today uses CRISPR-Cas9, a technology many of you have probably heard about. Think of CRISPR as molecular scissors guided by a GPS system. The GPS is a guide RNA that directs the Cas9 enzyme, the scissors, to a specific DNA sequence where it makes a precise cut. When this cut happens in a cell, one of two things typically occurs. Either the cell repairs the cut imperfectly, usually resulting in a non-functional gene. That's what we call a suppression drive, because it can potentially suppress or eliminate a population by disrupting essential genes. Or the cell can repair the cut using a template, and this is where the magic happens. In a gene drive, we can provide that template containing the gene drive machinery itself. So when the repair happens, the organism's DNA not only fixes the cut, but copies the entire gene drive into that location. Now both chromosomes carry the gene drive, and when this organism reproduces, nearly all of its offspring will inherit it. There are different types of gene drives being developed. Suppression drives, as I mentioned, aim to reduce or eliminate a population by targeting genes essential for survival or reproduction. In mosquitoes, for example, researchers have created suppression drives that target female fertility genes. The result? A rapidly collapsing mosquito population. Modification drives, on the other hand, don't aim to eliminate a species, but instead add or alter a specific trait. For instance, researchers are developing modification drives to make mosquitoes resistant to the malaria parasite. They'd still exist, but could no longer transmit the disease. We're also seeing the development of threshold-dependent drives, which require a certain release frequency to spread, versus self-sustaining drives that can spread even from a small initial release. These different approaches give us options depending on the specific application and risk considerations. Back in the 1990s, when I worked in the Wimmer lab at Stony Brook University, I had my own precursor to the gene drive that I conceptualized. It was a concept I called time bomb genes. It was a protein timer which would use mitochondrial DNA for expression. Mitochondrial DNA was proposed as it would only use maternal inheritance that would sidestep the genetic recombination challenges of nuclear DNA and provide a more stable platform for generational counting mechanisms. Additionally, targeting mitochondrial function would offer a powerful mechanism since energy production disruption would affect all cellular processes. This proposed synthetic mitochondrial protein would contain multiple cleavage domains that are processed in sequence with each generation. Each generation would be engineered to have cleavage sites that would be cleaved by specific proteases expressed during oogenesis. With each generation, one domain would be cleaved off. After a predetermined set of generations, the final cleavage exposes a domain that would upregulate a gene or cascade of genes that would disrupt mitochondrial function, killing all organisms containing the time bomb genes, which would have spread within the wild populations. With releasing around 10,000 initially genetically engineered female mosquitoes, after 3 generations, there would be more than 1 million mosquitoes carrying the time bomb gene complex, which is fine for local control and initial testing of the strategy. With releasing around 10 million genetically engineered female mosquitoes after 4 generations, it would be more than 1 billion mosquitoes which would be affected, which would be more than enough for many countries. With releasing around 2 billion genetically engineered female mosquitoes in various parts of the world, particularly sub-Saharan Africa and Asia, after around 15 generations, it would be enough to wipe out every genus Anopheles mosquitoes which carry malaria off the face of the earth. You would follow this up by releasing sterile male mosquitoes to the population, which would crash any straggling pockets of populations in the aftermath. You could always just continue to release more female mosquitoes in subsequent waves to decimate the populations further until it drops to 0. Of course, we didn't have the technology at the time to achieve this, and now there are simpler methods to accomplish this. At this point, you could just CRISPR such an engineered synthetic gene, encoding this protein into the mitochondrial genome, which is still difficult, but it's getting better, where the protein would include domains that inhibit toxic function until the final cleavage event, where it kills all organisms containing the complex, wiping out generations at a time. So in the end, I wasn't able to build the time bomb genes due to the technology available at my time. So, no Nobel Prize for me. I just got to be a crazy scientist coming up with fictitious scenarios on wiping out entire global insect populations every time I got bit by a damn mosquito or found a tick crawling up my leg. Building effective gene drives comes with significant technical challenges. One major issue is resistance, mutations that allow organisms to escape the gene drive mechanism. Just as bacteria develop resistance to antibiotics, populations can develop resistance to gene drives as well. Researchers are developing strategies to delay or prevent resistance, like targeting multiple genes simultaneously. Another challenge is confinement. How do you test something designed to spread rapidly without risking unintended release? Scientists are working on laboratory safeguards, including physical containment, molecular confinement strategies, and reversible gene drives that can be overwritten if needed. The science of gene drives is evolving rapidly, with new refinements and approaches constantly emerging. But what's clear is that we now have the technical capability to fundamentally alter how genetic inheritance works in wild populations, a power that demands careful consideration of where and how we might use it. So where exactly are we in the development of gene drives, and what applications are the closest to real-world implementation? Let's take a look at the current landscape. The most advanced work is happening with Anopheles mosquitoes, the primary vectors for malaria. Teams like Target Malaria, a research consortium funded by the Bill and Melinda Gates Foundation, and others have made remarkable progress. They've demonstrated gene drives in the lab that could either suppress Anopheles populations by targeting fertility genes or modify them to resist the malaria parasite. In 2024, we saw a major advancement when researchers demonstrated gene drives in Culex quinquefasciatus mosquitoes, vectors for West Nile virus, lymphatic filariasis, and other diseases. This was significant because it extended gene drive technology to a new mosquito genus, showing a potential broader applicability. The results from these lab studies have been striking. Some gene drives have shown inheritance rates above 90% and have been able to completely crash laboratory mosquito populations in 8 to 12 generations. Field case trials, the next step before actual field releases, are being planned in various locations, including Burkina Faso. But it's not just about mosquitoes. Researchers are exploring gene drives for a variety of other applications. In conservation, gene drives show promise for controlling invasive species that devastate island ecosystems. The G-BIRD Consortium, Genetic Biocontrol of Invasive Rodents, is developing gene drives aimed at humanely reducing population of invasive mice and rats that threaten endangered birds and other wildlife on islands worldwide. Since these island populations are naturally isolated, they provide a relatively contained testing ground. Agricultural applications are gaining attention too. Scientists are exploring gene drives to control crop pests like fruit flies and moths that cause billions of dollars in damage annually. There's also interest in using gene drives against weeds that have developed resistance to herbicides, potentially reducing the need for chemical pest control. And in public health, beyond malaria, researchers are looking at gene drives for ticks that spread Lyme disease, flies that transmit sleeping sickness in Africa, and even invasive mosquito species in regions where they weren't historically present, like Aedes aegypti in California. What's particularly exciting is how quickly this field is developing. The first proof of concept gene drives were created less than a decade ago, and we're already seeing sophisticated systems being tested in increasingly realistic conditions. For instance, researchers at Imperial College London have developed gene drives that can work even in genetically diverse wild mosquito populations, a crucial advancement since lab strains don't fully represent the genetic diversity found in nature. Scientists are also making progress on making gene drives more controllable. Some newer designs include genetic breaks that limit spread beyond a certain number of generations or geographic boundaries. Others can be deactivated by exposing the organism to specific compounds, giving us a potential off switch. While we're still years away from widespread deployment, the progress is undeniable. Initial field trials, likely in isolated settings like islands or clearly defined ecological zones, could begin within the next few years, pending regulatory approval and community consent. And just recently, researchers published results in Nature Communications showing that gene drives could be effective in reducing malarial transmission across various ecological conditions in West Africa. The models demonstrated that even accounting for variations in mosquito populations and environmental factors, gene drives could substantially reduce vector abundance and malarial prevalence. The research pipeline is robust, but moving from lab to field implementation involves not just technical hurdles, but also regulatory, ethical, and social considerations, which we'll explore in depth later this episode. Now let's shift gears a bit and talk about something that doesn't get enough attention in these discussions, the actual manufacturing challenge of producing gene drive organisms at scale. Because if we're ever going to implement these technologies in the real world, we need robust systems for production, quality control, and distribution. This is where manufacturing execution systems, or MES, becomes particularly relevant. Creating gene drive organisms isn't like traditional pharmaceutical manufacturing, where you're producing identical chemical compounds. We're talking about living organisms that need to be genetically consistent, viable, and capable of reproducing in the wild. The scale required is staggering. For mosquito control programs, we potentially need to produce millions or even billions of modified insects. A citywide release could require tens of millions of mosquitoes, and nationwide programs could scale that up dramatically. Here's where cloud-based manufacturing execution system platforms come in. These systems can track every aspect of the production process, from initial genetic modification to rearing to release. They create a complete audit trail that's critical for regulatory compliance and ensuring quality at every step. Think about the complexity. You need to monitor genetic stability across generations, ensure proper animal husbandry conditions, track performance metrics, and maintain detailed records for regulatory authorities, all while scaling production at levels never before attempted with genetically modified organisms. The manufacturing process itself faces unique challenges. Traditional bioreactors aren't designed for rearing insects or other complex organisms. Companies are developing specialized equipment, essentially automated insect factories with precise control of temperature, humidity, feeding, and other parameters. For example, biotech companies working on mosquito control are creating facilities capable of producing millions of mosquitoes weekly. These operations more closely resemble high-tech agricultural facilities than traditional pharmaceutical plants. Some use automated trays and conveyor systems to move mosquitoes through different life stages, with computer vision systems monitoring development. Quality control takes on new dimensions with living organisms. You need to verify not just the presence of the gene drive construct, but its stability across generations, the fitness of the organisms, and their ability to compete with wild populations. This requires sophisticated genetic testing, phenotype assessment, and even behavioral analysis. For example, if you're releasing male mosquitoes that carry a suppression drive, you need to ensure they're actually competitive for mating with wild females. A gene drive that doesn't spread is useless, regardless of how well designed the genetic construct is. Contamination control is another critical factor. Traditional biocontainment focuses on keeping organisms in, but with gene drive manufacturing, we need to prevent wild organisms from getting in and potentially introducing genetic variation that could compromise the gene drive's effectiveness. All of this requires sophisticated digital infrastructure. Cloud-based MES platforms provide real-time monitoring, predictive analytics, and the ability to quickly identify and address issues in the production process. They also enable remote monitoring and management, important when facilities might be located in disease-endemic regions far from company headquarters. Traceability is paramount too. Regulatory authorities and communities will demand comprehensive data on what organisms were released, where, when, and how they were produced. MES systems create this digital thread, linking every organism back to its production batch and genetic lineage. As we scale from lab to field, these manufacturing considerations become increasingly important. A gene drive that works perfectly in a small-scale lab production might face unexpected challenges when scaled to millions or even billions of organisms. Having robust manufacturing systems in place is crucial for identifying and addressing these challenges. The good news is that many of these technologies needed already exist in adjacent industries. Automated insect rearing systems have been developed for sterile insect technique programs. Biomanufacturing has advanced tremendously with cell and gene therapies, and digital manufacturing platforms like those we develop at Apprentice.io are ready to tackle these unique challenges. When we talk about releasing gene drive organisms into the wild, we're potentially altering ecosystems that have evolved over millions of years. So let's examine the ecological questions this raises, which are some of the most complex aspects of gene drive technology. First, there's the question of niche replacement. Nature bores a vacuum, as they say. If we successfully suppress or eliminate a species like the Anopheles gambia mosquito in an area, what might fill its ecological niche? While adult female Anopheles may feed on human blood, their larvae are aquatic and serve as a food for fish and other predators. Males feed on nectar and serve as pollinators for some plants. Would other mosquito species, possibly carrying different diseases, expand to fill this gap? Would there be cascading effects throughout the food web? The honest answer is, we don't fully know, but ecological modeling suggests the impacts might be less dramatic than you might think. Mosquitoes aren't keystone species in most ecosystems. While they serve as food for some predators, they typically don't make up a large enough portion of any predator's diet to be essential. In one study, researchers found that when they eliminated mosquitoes from test plots, Predator populations adjusted without significant disruption. Nature has considerable redundancy and resilience. That said, we're talking about potentially making changes across vast regions, and our ability to predict all ecological interactions is limited. This is why researchers are investing heavily in ecological modeling and conducting confined field trials before any wider release would be considered. Another key consideration in evolutionary responses, nature doesn't sit still, it adapts. With gene drives, we need to consider how target organisms might evolve resistance, how their behavior might change in response to selective pressures, and how other species in the ecosystem might adapt to these changes. For instance, if we deploy a gene drive that makes mosquitoes resistant to the malaria parasite, the parasite itself will be under intense selective pressure to overcome this resistance. It's an evolutionary arms race, and we need strategies to stay ahead. Geographic containment is another major question. Gene drives won't respect political boundaries or regulatory jurisdictions. A gene drive released in one country could potentially spread to neighboring nations or even continents depending on the organism's mobility and habitat range. Some species, like the Anopheles coluzi mosquitoes, are relatively contained to specific regions, while others, like the Aedes aegypti, have spread globally. Understanding these movement patterns is crucial for predicting how gene drives might spread. This is why researchers are developing gene drives with built-in geographic limitations. These local drives are designed to spread within a limited area and then stop, either because they've reached a population with a genetic barrier or because the drive itself has built-in limitations. Another approach is threshold-dependent drives that need to reach a certain frequency in a population to spread effectively. These would naturally fade out in areas where they don't reach this threshold, providing a form of inherent containment. How predictive can these ecological models really be? This is perhaps the most challenging of questions. We have sophisticated computer models that incorporate what we know about species interactions, population dynamics, and genetic factors. But ecosystems are fantastically complex, with countless interdependencies that we don't fully understand. That said, our modeling capabilities are improving rapidly. Modern ecological models can incorporate data from satellite imagery, climate patterns, human population densities, and genetic surveys to make increasingly accurate predictions about how gene drives might behave in specific environments. For instance, recent models of gene drive mosquitoes in West Africa incorporated factors like seasonal rainfall patterns, local mosquito population structures, and human settlement patterns to predict how effective gene drives would be at reducing malarial transmission. The key is taking an incremental, evidence-based approach. This means starting with laboratory studies, then moving to contained field trials, small-scale open releases, and only then considering wider deployments, with comprehensive monitoring at each stage to validate our models and catch any unexpected effects early. Lastly, we need to consider the time dimension. Gene drives don't just create a one-time change, they potentially alter evolutionary trajectories. We need to think not just about immediate effects, but impacts over decades or even centuries. This requires long-term monitoring programs and adaptive management strategies that can respond to changing conditions. As one ecologist put it, we're not just releasing an organism, we're releasing a process. That perspective demands both humility about our predictive capabilities and commitment to careful, staged implementation with robust monitoring. Now we come to perhaps the most challenging aspect of gene drive technology, the ethical questions it raises. These aren't just technical problems to solve, but profound questions about our relationship with nature, global decision-making, and intergenerational responsibility. First, whose choice is it to release a gene drive? If a gene drive could potentially spread across national boundaries, affecting ecosystems and peoples who had no say in its development or deployment, how do we ensure proper consent and participation in decision-making? Traditional models of governance and consent are challenged by technologies that don't respect borders. If country A releases a gene drive to combat malaria, but the modified organisms could potentially spread to countries B, C, and D, do all these nations need to agree? What if one refuses? This brings up complex questions of sovereignty and global governance that we haven't fully resolved for many transboundary issues, from climate change to internet regulation. With gene drives, these questions become even more acute because of the potential permanence of changes. Indigenous perspectives are particularly important here. Many indigenous communities have deep, generations-long knowledge of local ecosystems and cultural or spiritual connections to species we might modify. Yet these communities are often marginalized in global scientific and policy discussions. For example, in discussions about potentially using gene drives to control invasive mice in Hawaii to protect native birds, Native Hawaiian perspectives on both the invasive species and the intervention itself are essential to consider. Their cultural relationship with the land, or Aina, provides insights that Western scientific approaches might miss. Religious and cultural viewpoints also vary widely on genetic intervention. Some religious traditions emphasize humanity's roles as stewards of creation with responsibilities to alleviate suffering, which might support interventions against disease vectors. Others emphasize humility and caution about altering nature, particularly in permanent ways. These perspectives aren't just abstract considerations, they shape how communities will respond to proposed gene drive releases and whether they'll trust the science behind them. Perhaps the most challenging ethical question is the balance between action and inaction. Not using gene drives also has moral consequences. Malaria still kills over 600,000 people annually, mostly children under 5. If we have the technology that could dramatically reduce this burden, do we have an obligation to use it? As bioethicist Arthur Caplan puts it, the notion that nature is benign and always to be preserved exactly as it is does not match the reality of diseases that kill and disable millions of people, especially children in poor countries. This forces us to weigh risks against benefits, known harms against uncertain ones, present suffering against potential future consequences. And these calculations look very different depending on whether you live in a malaria endemic region or not. Then there's the question of dual-use concerns, the potential for technologies developed for beneficial purposes to be misused. Could gene drive technology be weaponized? Could it be used to create economic havoc by targeting agricultural species? The technical barriers to creating a malicious gene drive are still significant, but they're lower than for many other potential bioweapons. This requires both technical safeguards in the design of gene drives and governance frameworks that prevent misuse while enabling beneficial applications. It's a delicate balance that the scientific community and policymakers are actively working to strike. Finally, there's the matter of who benefits financially from gene drive technology. If a gene drive is developed to combat a tropical disease, but the patents and profits flow primarily to institutions in wealthy countries, are we reinforcing existing inequities even while addressing disease burden? These ethical questions don't have simple answers, but they demand thoughtful engagement from diverse perspectives. The decisions we make about gene drives will reflect not just scientific assessments, but our values as a global community. What's encouraging is that these ethical discussions are happening alongside the technical development, not as an afterthought. Organizations like the WHO and scientific consortia like Target Malaria have emphasized the importance of community engagement, ethical oversight, and inclusive decision-making from the earliest stages. So how do we actually govern our technology with the power and reach of gene drives? This question is keeping regulators, policymakers, and scientists up at night, and with good reason. First, let's acknowledge that our existing regulatory frameworks weren't designed with gene drives in mind. Most countries regulate genetically modified organisms through agriculture or environmental protection agencies. These frameworks typically assume that GMOs won't persist in the environment long term or spread beyond their initial release area, exactly the opposite of what gene drives are designed to do. Take the Cartagena Protocol on Biosafety, which governs international movements of living modified organisms. It was designed with conventional GMOs in mind and lacked specific provisions for gene drives that could cross borders through natural organism movement rather than human transport. The result is a regulatory patchwork that varies widely by country and doesn't fully address gene drives' unique characteristics. Researchers and developers find themselves navigating uncertain requirements, sometimes delaying progress on potentially beneficial applications. International governance faces even greater challenges. No single global body has clear authority over gene drive releases. The Convention on Biological Diversity provides some framework, but enforcement mechanisms are limited, and notably, the United States isn't a party to this convention. Various international organizations are trying to address this gap. The World Health Organization has developed guidance specifically for testing genetically modified mosquitoes. The IUCN, International Union for Conservation of Nature, has established principles for using gene drives in conservation, and the UN Convention on Biological Diversity has called for broad international consultation before any releases. But implementing these guidelines requires cooperation across nations with very different resources, technical capabilities, and priorities. A country suffering from high malaria burden might view the risk-benefit calculation very differently than a country concerned primarily with biodiversity protection. Public engagement presents its own challenges. How do you meaningfully engage communities on a technology as complex as gene drives? Technical literacy varies wildly, and conveying both the potential benefits and risks in accessible terms is difficult. Some projects, like Target Malaria, have pioneered approaches to community engagement in potential release areas. This includes everything from village-level discussions to training local community members as liaison officers who can explain the technology in local languages and cultural context. This concept of community consent raises complex questions too. What constitutes a community when a technology could potentially affect entire ecosystems or regions? Who represents that community? How do you balance the local perspectives with broader national or global interests? Various models of governance have been proposed. One approach is phased testing, starting with laboratory studies, then contained field trials, limited field releases, and only then considering wide-scale implementation. Each phase would require specific approvals and evidence of safety from the previous phase. Another model emphasizes transnational governance networks that bring together scientists, regulators, affected communities, and other stakeholders across national boundaries to make collective decisions about gene drive releases. Then there's the crucial question of safeguards and kill switches. Can we truly control gene drives once they're released? Scientists are developing several approaches. There are self-limiting gene drives designed to lose effectiveness after a certain number of generations. Split drives separate the CRISPR components so that the drive can't spread unless all the parts are present. And reversal drives can potentially overwrite or neutralize an earlier gene drive. But we need to be honest, none of these safeguards are foolproof. Once genetic changes spread through wild populations, recalling them completely may simply be impossible. This reality doesn't mean we shouldn't proceed, but it demands extraordinary caution and thorough testing of safeguards before any environmental releases. Monitoring is another critical component. If we do release gene drive organisms, how will we track their spread, ecological impacts, and effectiveness? This requires not just technical capability, but sustained funding and institutional commitment over decades, timescales that typically exceed research grants or political cycles. As we navigate these governance challenges, transparency is essential. The scientific community has largely embraced this principle, with researchers publishing their gene drive work openly and engaging in public discussions about risks and benefits. The transparency builds trust and allows broader participation in shaping how this technology develops. It also enables early identification and potential issues that researchers themselves might not have considered. As we wrap up today's exploration of gene drives, I want to take a moment to look toward the horizon to consider where this technology is heading and how we might navigate this path responsibly. The next generation of gene drives is already taking shape in research labs around the world. Scientists are developing drives with unprecedented precision and control mechanisms. For instance, daisy chain drives split the system into multiple components that lose elements over generations, causing the drive to naturally fade out after a predetermined spread. This could allow for temporary interventions rather than permanent genetic changes. Other researchers are working on drives that respond to specific environmental triggers, activating only in certain geographic areas or under specific conditions. Imagine a mosquito drive that only functions in areas with high malaria transmission, automatically deactivating if it spreads to regions where the disease isn't endemic. Robust safeguards are evolving alongside the technology itself. Beyond the technical kill switches we discussed, researchers are developing ecological and geographic containment strategies, testing gene drives on islands or in isolated areas before considering broader releases, for example. They're also creating multiple redundant safeguards, recognizing that no single approach is infallible. But perhaps the most important safeguard is the one we're engaging in right now: open, transparent, and inclusive dialogue. The scientific community working on gene drives has, to its credit, been remarkably transparent about both the promise and pitfalls of this technology. This discussion has expanded far beyond laboratory walls to include ethicists, ecologists, social scientists, policy experts, and affected communities. This transparency serves multiple purposes. It builds trust, prevents the perception of scientists working in secret on technologies with global implications, and brings diverse perspectives that strengthen both the technology and its governance. It also allows for broader societal reflection on questions that transcend science alone. Questions about our relationship with nature, our responsibilities to future generations, and our values as a global community. As we look to the future, I believe the path forward lies in balancing innovation with responsibility. Gene drives offer extraordinary potential benefits, millions of lives saved from malaria, endangered species protected from invasive predators, reduced dependence on chemical pesticides. The possibilities demand our serious consideration and continued research. At the same time, the potential risks and uncertainties demand extraordinary caution. Unlike many technologies where we can iterate quickly and fix mistakes as they arise, gene drives could create changes that are difficult or impossible to reverse completely. This reality calls for a measured, staged approach to testing and implementation with comprehensive assessment at each step. I want to leave you with a message of hope and humility. Hope, because human ingenuity continues to develop tools that can address some of our most persistent challenges. The same creativity that developed gene drives is also creating the safeguards, governance frameworks, and ecological models that could allow us to use them wisely. Yet humility, because we're tinkering with systems of staggering complexity, ecological networks, and evolutionary processes shaped over millions of years. Our understanding, while growing rapidly, remains incomplete. This calls not for abandoning the technology, but for proceeding with appropriate reverence for what we don't fully comprehend yet. The story of gene drives is still being written, and importantly, we all have the opportunity to shape how it unfolds. Whether you're a scientist in the lab, a policymaker setting regulations, or a citizen engaging in public discussions, your voice matters in determining how we navigate this powerful technology. Perhaps the most hopeful aspect is that we're having these conversations now, before wide-scale releases, before crossing points of no return. We're collectively grappling with the ethical and governance challenges alongside the technical ones, an approach that offers our best chance to realizing the benefits of gene drives while minimizing their risks. So, as we move forward, let's continue this dialogue. Let's bring diverse perspectives to the table, commit to evidence-based decision-making, and hold ourselves to the highest standards of responsibility. Gene drives are a technology that shows us both the extraordinary power of human innovation and our profound responsibility as stewards of our planet's future. The choices we make about gene drives won't just affect our generation, but potentially all generations to come. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.